All right, so let's say I have this really long molecule of DNA and I don't want to analyze the entire molecule of DNA. What I want to do is I want to isolate and amplify a particular region of interest. By amplify, I just mean make a whole bunch of copies. And let's say I want to make a whole bunch of copies of this specific region because this is the region that contains a short tandem repeat. Well, based off of what we know about DNA replication, there's a few things that I'm going to need in order to replicate this region of interest. The first thing I need are some deoxyribose nucleotide triphosphates, like ATP, TTP, CTP, GTP. The next thing I'm going to need is maybe some helicase, because I need to unzip the DNA in order to begin to make copies of it. But I actually don't need helicase, because what I'm going to do instead is I'm going to run this replication inside of a test tube, and I'm going to use high temperatures. I think it's like 95 or maybe 96 degrees Celsius. I'm going to use high temperatures to denature the double-stranded DNA. And so what that means is just that I'm separating the two strands. I'm breaking apart the hydrogen bonds. I'm not actually destroying the sugar phosphate backbone. I'm just breaking the hydrogen bonds, holding the double strands together, which is normally what helicase does, so I don't need helicase. I'm just going to use really high temperatures. Now the other thing I need is DNA polymerase, right? DNA polymerase is responsible for actually synthesizing new DNA, right? It's creating the daughter strand by reading the template strand. And I'm going to use a special type of polymerase. This comes from a bacteria cell because it needs to be able to withstand the very high temperatures, right? 95 degrees Celsius, that's almost boiling. You try to take a human DNA polymerase and expose it to those temperatures, it's not going to do anything because it's going to unfold. Now, if you remember from a few days ago when we were talking about DNA replication, DNA polymerase cannot synthesize nucleic acids without having some sort of primer to work off of, or without having some sort of 3' end of a pre-existing nucleic acid strand to work off of. So based off of that logic, you might think that we need to add some primase. But we actually don't need to add primase, because what we can do instead is we can just straight up add DNA primers into the solution. And these DNA primers are super important because these are essentially going to tell DNA polymerase where along the DNA molecule to do replication. The primers are actually flagging the region that needs to be copied. And so let's zoom in on this region of interest. Let's look at the DNA molecule and the actual base pairs that flank the region of interest, right? So let this red region represent TCAT, right? So this individual has 2 repeats of TCAT. The complementary strand would be AGTA, and so I want to isolate and amplify their short tandem repeat, TCAT. I want you to pause the video and think about and try to write down, identify what primers would I need to use in order to tell DNA polymerase to begin making copies of this region of interest. All right, I'm going to pause my video and kind of get my drawing set up. You do the same and see if you can identify. Let's make the primer, I don't know, 10 base, or not base pairs, just 10 nucleotides long. 10 nucleotides long. All right, I'm pausing my video. Okay, so I've separated my DNA molecule using high temperatures. I'm going to let the solution cool down, like the video says, to allow the primers to anneal. That just means form hydrogen bonds with these exposed strands. And so there's a couple different places. I could have my primer stick to the DNA. I can have a primer stick here, and there would need to be a 3' end right here because I'm going to want to copy to the right. Or I could have a primer, I could lay a primer down right here, and again, this would need to be a 3' end. And in this case, I would want to be copying to the left. I could have a primer down here with a little 3' poking out because I'd want to copy to the right. Or I could have a primer here with a little 3' poking out, right? If we're using my arrow convention from a few days ago, it would need to look like that, right? And we would want to be copying this way. So these are 4 possibilities, but there's only 2 that are actually realistically possible, because there's only 2 of those 4 configurations that are following the anti-parallel rule that we have to keep in mind. So for example, if I try to put a primer right here, right, think about the orientation of this primer. This primer is 5 on this side and it's 3 on this side. That's not going to work because this primer would be parallel to the template strand, not anti-parallel. So that's not going to work. So let's try putting a primer down on the opposite side. Let's do 5, 3. Does that work? Yes, it does, right? You have the primers anti-parallel to the template strand, and the 3' is right here, so DNA polymerase can click in and begin synthesizing towards the left, making a copy of that region of interest. Now it's also going to be making copies of parts of DNA that we're not so interested in. You don't have to know the specifics, but by the 3rd round of replication, DNA polymerase will be exclusively making copies of just the short tandem repeat, of just the region of interest. So it takes a couple rounds, the point is that it takes a couple rounds of replication before we actually isolate the region of interest. But once we isolate it after the 3rd round, the constant, or the amount of the region of interest, grows exponentially in our test tube. And within something like 24 hours, we've got millions of copies of the target sequence. Okay, so what is, I said 10 nucleotides long, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? And so we would say 5, and then 3. And then down here, I would need to have, let's see, 5, 3. Is that anti-parallel? Yes, it is, to this template strand, right? This is my template strand now, right? If I try to do it here, right, 5, 3, it's not anti-parallel to the template strand. I've got my 5s on the same side, I've got my 3s on the same side. Okay, so I'm going to put a primer down here. I'm going to go ahead and pause the video so I can put my primer down. All right, so this is what my primers need to look like. That's going to allow DNA polymerase to click in and replicate. It's going to click in on the top and replicate. And if I just keep doing this over and over and over again, like I said, I do this overnight, right? I do this in the afternoon before I leave the lab for the evening, come back the next morning, and bam, I've got millions of copies of this STR. And this is happening, well, on one piece of DNA, but keep in mind if we're collecting this DNA from an individual, or it's DNA found at a crime scene, we're actually going to be making sorry, lost my train of thought. We're actually going to be making 2 fragments, right? Because the individual's diploid, right? So we're going to be making this STR, which is 2 repeats of TCAT, and then depending on what their other allele is for the STR, we'll be making copies of those as well, because the primer works for both pieces of DNA, for both the maternal and the paternal chromosome, right? So after 24 hours of letting this ride, I'll have millions of copies of these, right? And so these are going to be what, TCAT is 4, and it repeated twice, so 8. This is going to be about 8 base pairs long. It'll actually be a little bit longer because we'll have, well, never mind. It could be a little bit longer, but we'll just say 8 base pairs long. And then let's say they are not homozygous. Let's say they're heterozygous. So this one looks like it's about, I don't know, maybe 3 repeats. So 12 base pairs long. And so, you know, this is all existing inside of a test tube. Right here I've got a little vial, and I've got all of these, you know, fragments that are 8 long, and then I've got all these fragments that are 12 long, and of course I can't see it with the naked eye. It just looks like a clear solution, right? But then what I'm going to do is let's look at it from the side, and then I'll draw it from the top. This video is almost done, right? I'm going to dump all of that into my well. I'm going to flip the switch. I'm going to run a current through the gel. Here I'm going to have the negative side close to the well. I'm going to have the positive side further away from the well, and the DNA molecules are going to migrate through the gel. I'm going to run this for about 45 minutes to an hour, and when I turn the power supply off and stain the gel with ethidium bromide and x-rays, I'm going to see something like this, right? And so if we look at it from sort of a bird's eye view, it'll look something like this. I'll see a band here, and I'll see a band here, right? And so this band right here is going to be my shorter, my 8 base pair fragments that were amplified. And then this band right here is going to be my 12 base pair fragments. Okay, and so all you're doing in today's activities is you're going to look at a case of our druid Dracula, our individual who was trying to learn how to become a vampire, and you'll see how that turned out for him. And then you're going to be looking at a case study where you're trying to reconstruct the family tree of an Egyptian family. So yeah, that's your activity for Wednesday. You guys are coming up on a nice, 5-day break. It's going to be a break for me too. This is taking a lot of time. I should be asleep right now, but regardless, it'll be a nice break for all of us. And yeah, enjoy it, and I'll see you towards the end of February, beginning of March.